Hi, this podcast is on Mendelian genetics, and this is actually an introduction to genetics. In our introduction to genetics, we'll start with a talk about Gregor Mendel. Gregor Mendel, 1822 to 1884. He was an Austrian monk, and Gregor Mendel studied the inheritance of traits in pea plants. I'm not going to go into a lot of detail about Gregor Mendel. You have another video in this lesson to watch. Mendel's work, the important thing, was not recognized until the turn of the 20th century. And in fact, Mendel didn't know anything about DNA, and he knew that there was some sort of a particle responsible for inheritance. He just could not, the technology wasn't there to understand DNA. But between 1856 and 63, cultivated and tested about 28,000 pea plants. And what he found from his pea plant experiments was that the plant's offspring retained traits of the parents, and this is the basis to genetics, which is why to this day Gregor Mendel is called the father of genetics. So Gregor Mendel stated that physical traits are inherited as particles. Now remember that Mendel didn't know that particles were actually chromosomes in DNA. He said that something was being inherited, that some sort of a particle was being inherited from parents to offspring. So a little bit of gene terminology to get us going with genetics. A trait is a characteristic that can be passed from parent to offspring. Heredity is the passing of traits from parent to offspring. We have some types of crosses. We have a monohybrid cross, which is a cross involving a single trait, for example, flower color. We have a dihybrid cross, which is a cross involving two traits. So for example, flower color and plant height. Punnett squares. Punnett squares are an important part about genetics as they're used to help solve genetic problems. And we'll be going into more detail on Punnett squares when we look at the various crosses. But as a basics, you can read through this to sort of understand. The only thing I'd caution you on with this diagram and this example, while I like what they sort of talk about in terms of how to construct Punnett squares and how to figure out some meanings to them, I would caution you on using letters like Y's, where the big letter and the lowercase look very similar. I would sooner choose A's and R's and B's, anything where the uppercase and lowercase are different. But the fundamentals of the Punnett square are really good with this slide, so please read through it. So alleles. Alleles are actually a form of a gene, and alleles can be either dominant or recessive. If an allele is dominant, it's stronger of the two genes. It's expressed in the hybrid, and it's represented by a capital letter R. If an allele is recessive, it shows up less often in a population, and it's represented by a lowercase letter r. A little bit more terminology here, the genotype. The genotype of an organism is the gene combination, and we represent genotype with letters. Now remember what these letters represent. If I look at this capital R, it represents a trait in which there are two possible alleles, and both of those alleles happen to be dominant. Now remember that we're really looking at those homologous chromosomes if we think back to meiosis. Now the phenotype is a little different. The phenotype is the physical features. So in other words, the phenotype is what we see when we look at a trait. So for example, if this was a trait for flower color and this was a big R, this may be the genotype of the organism, but we may see red. Same with this one here. If we were to look at this genotype here, big R, little r, this is a heterozygous. And so we're still seeing red as a phenotype. And this one would be the only phenotype in which we'd see white flowers. So you really need to know the difference between genotype and phenotype. So a little bit more about genotype and phenotype in flowers. Capital R is representative of red flowers. Little r is yellow. All genes occur in pairs. And so two alleles will affect the characteristics of an individual, and possible combinations, like we said, are big R, big R, big R, little r, little r, little r. Homozygous dominant, heterozygous, and homozygous recessive. So the homozygous genotype is a gene combination involving two dominants or two recessive genes. This is also called purebred. Mendel didn't refer to them as homozygous. Mendel referred to them as purebred. The heterozygous genotype is a gene combination of one dominant but also a recessive allele, and Mendel referred to these as hybrids. So Mendel's pea plant experiments were crucial in the understanding of genetics and how traits were inherited. And so Mendel used the pea plant for a variety of reasons. One, it can be grown in a small area like a monastery. It produces lots of offspring, if you imagine pea pods. They produce pure plants when allowed to self-pollinate several generations. So pea plants can self-pollinate, and they can be artificially cross-pollinated. And Mendel did this very laboriously. Now, just a little bit of reproduction in flowering plants. Pollen is what will contain the sperm. It's produced by the stamen. So if we look at our flower here, that's a stamen. And that's where the pollen is located. The ovary contains the egg. So here's the ovary. And that's found inside the flower. Pollen will carry sperm to the egg for fertilization in one of two ways. A plant can self-fertilize, meaning it can occur in the same flower. So pollen from the same flower can pollinate that flower, or cross-fertilization between flowers. And obviously cross-fertilization is going to allow for a little bit more genetic variability than self-fertilization. So what Mendel did was he hand-pollinated flowers using a paintbrush, and this is something similar. He snipped the stamens of the flowers to prevent self-pollination. He covered each flower with a cloth bag so that pollen would not contaminate them. And what he did with this experiment is he traced traits through several generations by methodically selecting for what he was looking for. So here's how Mendel began. He took a self-pollinating pea plant. He allowed fertilization to happen. He traced or allowed that flower to come to fruit, so to make seeds. And then he planted those seeds to allow germination to take place. And Mendel actually didn't just stop with one trait. Mendel looked at 8 different pea plant traits in his crosses, and this is a list of them. I would never ask you to memorize a list, but I mean, have an idea of some of the traits that Mendel looked at. And so again, this is a little bit more of a diagram showing what these traits actually look like in the peas. So seed shape, seed color, pod shape, being inflated or wrinkled. And these may be things that you've already noticed in your own gardens. Flower color, purple or white. Flower positions, they can be on the stem or they can be at the tip. This is called a terminal. This is axial. Stem length can be tall, and plants can be short or dwarf. So Mendel's experimental results. After doing all of those plants, Mendel got the following sort of ratios, which he was very happy of, because you can see the pattern that started to emerge. Now this is the F2 generation, and we'll talk a little bit more about F1 and F2 and what those mean. But essentially, after Mendel allowed the offspring of his first cross to mate, he noticed that there was a 3-to-1 ratio of dominant phenotypes to recessive phenotypes. And this was very consistent with all the traits that Mendel looked at in the pea plant, which definitely suggested that there was something going on with how traits are being inherited. So now, this is just asking if the observed ratio matched the theoretical ratio. Well, Mendel expected a 3-to-1 ratio of dominant to recessive, and what he observed was 2.96 to 1. And you can see that that's very close, and the discrepancy is actually due to statistical error. And the larger we get the sample, the more the we get an approximate to the theoretical ratio. Now, just a little bit here on some generations. The parental generation, or the P1, that's the parental generation in a breeding experiment. So for Mendel, it was 2 homozygous dominant and a homozygous recessive pea plant for a particular trait. The F1 generation, called the first filial generation, is the first generation offspring. Now, the F1 comes about from the breeding between the P1s. So in Mendel's experiments, what he noticed was in his F1s that all of those offspring exhibited the dominant trait. The recessive trait almost disappeared. And of course, Mendel asked, well, where did the recessive go? And so he went one step further. He allowed those F1s to self-pollinate to produce the F2s. So the F2 generation is the second filial generation, and it's from breeding individuals within the F1, and that's in that F2 generation he saw that ratio. And this is this again. So if we look at the parentals, he crossed 2 pure plants. So they were looking at plant height. So he crossed a homozygous dominant tall and a homozygous recessive short. What he found was that they were all tall. They resulted in hybrids. And when he allowed those to sort of reproduce, he would get, on average, a ratio of 3 tall to 1 short. And that's our introduction to genetics.